Hello, AP Bio folks. Today we're going to continue talking about cell types and cell structures, kind of continuing off some of the ideas that we talked about last week and some of the things that you looked at yesterday. So we are officially entering Unit 4. The notes we look at today are in your Unit 4 note packet, though we will still be jumping around a little bit. So I'll try and kind of help you find where those notes are. But you might want to grab your Unit 4 note packet so you can follow along and add some notes as we go through. In your textbook, this unit covers Chapter 4 and part of Chapter 5 about the membrane structure and function. So that's where we are in the textbook. So yesterday we talked about the fact that cells are the basic structural and functional units of life. I asked you to draw a prokaryotic cell and a eukaryotic cell. So take a look at what you drew yesterday and see which of these things you included in your drawings and which ones you maybe didn't. There are a few things that all cells have in common. Every single cell is surrounded by a cell membrane, which is also referred to as a plasma membrane. They're filled with a fluid called cytosol. Now, you've probably heard the word cytoplasm. That's what we talk about the most in 10th grade. Cytosol specifically refers to the liquid. Cytoplasm actually refers to the liquid and all the organelles that fill the space of the cell. So you can use those two words kind of interchangeably, but cytosol is technically just that jelly-like liquid that kind of fills in the space within the cell. All cells contain genetic material. They all contain DNA, and they all have ribosomes. Every single cell, no matter what type, needs to make proteins that carry out a variety of functions within the cells, but those proteins are made by ribosomes. Ribosomes are not technically organelles because they don't have a membrane around the outside, so all cells, both prokaryotic and eukaryotic cells, contain ribosomes, those protein-making structures. But of course, there are some differences between these two cell types as well. Prokaryotic cells fall into two domains. Living things are divided into three domains. Two of them contain living things that have prokaryotic cells. Those domains are bacteria and archaea. In these cells, in prokaryotic cells, the DNA is not enclosed in a membrane, so they don't have a nucleus. The DNA is kind of centered within this region of a cell, though, and that region within the cytosol is called the nucleoid region. So the DNA isn't just totally loose within the cell. It is still kind of compact within one area that's called the nucleoid region. But there's no membrane around it. Prokaryotic cells also don't have membrane-bound organelles, but their cytoplasm might have regions that, say, have a concentration of a particular enzyme that carries out a chemical reaction or has a concentration of ribosomes that build proteins. So there might still be some different regions, but those regions are not separated by membranes. Prokaryotic cells also tend to be much, much smaller than eukaryotic cells. The eukaryotic cells all fall into one domain that's called eukarya because they have this in common. And eukaryotic cells have most of their DNA enclosed in a nucleus. So you'll notice this says most because some of the organelles actually have a little bit of their own DNA. The mitochondria contain a little bit of their own DNA, and so do chloroplasts if you're looking at a plant cell. We'll actually talk a little more about that over the next few days and why a couple of those organelles have a little bit of their own DNA. The cytoplasm in eukaryotic cells contains some membrane-bound organelles. So that's all the ones that you normally think of, like the mitochondria, the chloroplasts, the vacuoles, the Golgi apparatus, the endoplasmic reticulum. We'll be reviewing a bunch of those next week. But the point of those organelles that are separated by membranes is that provides little tiny microenvironments for specific metabolic functions. If you remember last week, you looked at that cell membrane packet. You worked through some problems that talked about compartmentalization. And it used the analogy of like organizing your bedroom by having closets and drawers and desks and boxes to kind of organize your stuff. Eukaryotic cells use membranes, these little sections, to separate different regions for different metabolic functions. So particular activities are happening within those different organelles. In general, eukaryotic cells tend to be much larger than prokaryotic cells. So these are the two main types of cells that fall into those three domains. One of the assignments you'll be working on over the next several days is to learn a little more about where these three domains of living things came from, and kind of the origins. We're going to continue that story of the origin of life. In the previous unit, we talked about the chemical origins of life and how the first biomolecules might have evolved and formed into simple cells. Now we're going to talk about how those cells became what we now know as prokaryotic and eukaryotic cells. So this assignment will be posted today, and this is one thing that you'll have to work on for the next few days, is to learn more about this. We'll come back to this topic next week. For today, we're going to talk about how all of these cells get the substances that they need in and out through their membrane. So we're going to jump ahead in your note packet again and move back to where we were talking about this idea of transport in and out of cells. You may remember this chart that we looked at before. There are lots of different types of molecules that cells need. All cells need to get things like, like oxygen and carbon dioxide that they need for their different metabolic processes. All cells, of course, need to take in water, and they need to get rid of wastes. Urea is one type of waste product. They need to take in molecules like carbohydrates to get energy. They need different types of ions that they use for a variety of functions. Some of these ions, remember, work as coenzymes and cofactors to help enzymes carry out chemical reactions. But because these molecules have different chemistry, they enter the cell in different ways. And again, we talked about this before, so I'm not going to go through all of these in detail. So today we're going to review a little bit more and go a little bit further with talking about transport. This slide is not in your notes. This is something I added today just to kind of review and summarize these different types of transport. Passive transport is the one that we're focusing on now, and that's the type of transport that happens without any energy because things are moving down their concentration gradient. So these are types of diffusion. Diffusion is when molecules move from where there's a high concentration of that molecule to a low concentration. So it's like rolling down a hill. It doesn't require any energy. Some molecules need a little bit of help in order to do this. Facilitated diffusion still does not require energy, but it does require a protein channel. Those proteins don't do anything, they don't change shape, but these are some of those proteins that go across the cell membrane, and their main function is to provide a chemically friendly pathway for those charged ions or polar molecules that don't want to go through the hydrophobic interior of the cell membrane. So it makes a hydrophilic pathway where those charged molecules can move through more easily. The third type of passive transport is osmosis, which is the diffusion of water. And like we mentioned before, that is usually facilitated diffusion. Some water can get through the lipid bilayer, but because of the hydrophobic interior, most of it moves through those aquaporin proteins that provide a hydrophilic channel. There is, of course, also active transport that requires energy to move big substances or to move them against the gradient. We're going to come back to active transport at the beginning of second trimester. Today, we're going to focus in specifically on osmosis. And this will tie back into the potato lab that we did last week. And you're going to be revisiting that over the next few days as well to go a little bit deeper in your understanding of what happened in that lab. Water moves in and out of cells for two main reasons. Actually, water moves everywhere. This also includes moving up through plants and other places. Water moves for two main reasons. One of them has to do with the solute concentration. And this is where that idea of hypertonic and hypotonic comes in. You started thinking about this already with the potato lab. We call the difference in solute concentration, we call that the solute potential. And there are some equations that go with this. You'll see this little symbol right here. This symbol, that's some Greek letter, and I honestly don't remember which one that is, but with a little S next to it, that stands for solute potential. And that's one of the reasons that water moves. When you add solutes, that actually decreases the water potential because the water kind of sticks to the things that are dissolved in it. It can't move as easily. So if you look at this diagram over here, the larger circles represent sugar molecules. And when you add more sugar into a solution, some of the water molecules, so my pen is red, but you have to imagine that these are water molecules. Some of those water molecules actually stick to that solute. That's part of the process of dissolving. And because it's stuck to the solute, it can't cross that membrane as easily. So water will move from where there's more free water to where there's less. And we're going to come back to that idea in another slide or two here. The other reason that water moves is because of differences in pressure. So again, you see this Greek letter symbol here with a little p. That stands for the pressure potential. And this one I think is easiest to explain if you just think about something as simple as like a tube of toothpaste. Where you are squeezing on it right here, there is really high pressure on that toothpaste. Out here in the room, there's no pressure. And so the toothpaste is going to move from where there's higher pressure to where there's lower. It's going to move out of the tube when you squeeze on it, right? That's the idea of pressure potential. These two together add up to water potential. And this, again, is an equation on your equations and formula sheet. Water potential is just equal to the pressure potential plus the solute potential. So those two combined make up this thing called water potential. And just like everything else, water is going to move from high to low. So it's going to move from where there's higher water potential to where there's lower water potential. There's another equation on your equation sheet for calculating solute potential. There are a few things that I want to highlight, particularly with this equation. Oops, there we go. So you can actually calculate the solute potential of a solution using this equation. All of these variables are defined on your equation and formula sheet, so you don't need to memorize any of this, but I do want to point out just a couple of things. First of all, notice there is a negative sign in front of this equation. That's really important because it's pretty small, so it's easy to overlook. But this is negative I times C times R times T. I is what's called an ionization constant. That's different for each substance. Your equation sheet gives you the ionization constant for sucrose, which is equal to 1, because sucrose doesn't ionize in water. So many of the problems that you might see with this equation will talk specifically about sucrose. So always remember to look at your equation sheet because it tells you that. If it's talking about something other than sucrose, this number will be given to you for that substance. The C stands for the molar concentration. So that's a number that will be given to you in the problem. In our potato lab, you will actually be figuring out the molar concentration of sucrose within your potatoes. Capital R is a pressure constant, and again, this is given to you, so you will always use this number for R, and it has this big, long, complicated unit. You don't need to worry about that. You just need to know that this is the number you always plug in for R. And T is temperature. The only important thing you need to notice is that the temperature must be in Kelvin. I believe it gives you this on your equation sheet as well. If the temperature is given to you in degrees Celsius, which it often is, make sure that you add 273. So this equation is not difficult, but it does have a couple little details that you just need to keep in mind as you calculate this. So the way that solute potential part works, because when we're looking at something like osmosis, we're really focusing more on the solute concentrations. Pressure doesn't make a huge difference if you're just talking about water moving in and out of a cell in most cases. Most of that movement is just due to solute concentration. When you add solute, it actually decreases the solute potential of water. Because like I said before, the water molecules that are bound to the solute can't cross the membrane. And so what you want to think about here is something called the free water concentration. In other words, the ability of water to move freely without being connected to something. So if you look at this picture here, this is showing a bunch of sodium chloride, that's salt, dissolving in water. And you can see as we add this dissolved substance, all of these water molecules, that's 3 water molecules around this chlorine, have formed hydrogen bonds to that negatively charged ion. So these water molecules are now not free to move across membranes as easily because they're bound to this charged ion, and that makes a much bigger clump of atoms that just can't freely pass through a membrane. And same thing over here. All of these are kind of bound together in this big clump. So everything I'm circling is no longer free water. And so when you add a solute like this, the amount of water that's actually free to move is much lower. The molecules that you have maybe over here on the side are still free and over here, but there's a much lower percentage of free water when you add a solute. And so if you think about that in terms of our hypertonic, hypotonic, isotonic solutions, it can help you kind of figure out which direction the water is going to move. So when a solution is isotonic, we'll start with that one because it's kind of the easiest one to visualize. Isotonic means there's equal concentration on both sides of the membrane. And so you do have some water. So my little red dots are going to represent water molecules in these drawings. You do have some water that's bound to the solute on both sides of the membrane. So these are water molecules bound to these solute particles. But you also have some free water in between, and the concentration's about equal. So if the solution's isotonic, the water is going to just randomly move across the membrane equally in both directions. So there's no net movement. Overall, it looks like nothing's really changing because the concentrations are staying the same. If you put that cell in a hypotonic solution, remember hypotonic solution means there's a lower amount of solute that's dissolved in the solution compared to the inside of the cell. So there's less solute out here, but that also means that there's less water molecules bound to the solute, so you actually have a lot more free water out here. Remember, my red dots are representing the water molecules. Oops, that one's floating through the membrane, I guess. Compared to inside the cell, you have a lot more water molecules that are bound to these solute particles. So there's a lower percentage of water that's just free to move. So even though there's still a lot of water in the cell, there's more free water out here and less free water inside. So the water is going to move down its concentration gradient like everything else. It'll move from where there's a higher concentration of free water to where there's a lower amount of free water inside the cell. So in a hypotonic solution, water will move inside of that cell and the cell will gain mass and expand. The opposite's true in a hypertonic solution. A hypertonic solution, remember, has a higher concentration of solute outside of the cell. And that means if there's a lot of solute out here, it also means a lot of the water molecules are bound to that solute. So there's a lot less free water just floating around in between here. Inside the cell, there's still a little bit of water bound to those solute particles, but there's a lot more free water. So again, water is going to move from where there's a higher concentration of free water to where there's a lower concentration of free water. So in a hypertonic solution, the cell is actually going to lose water and shrink and shrivel up. Living things need to be able to, this one first. Plants kind of take advantage of this fact to help get water into their cells. So root hair cells are kind of shaped like this. They're specialized. They have these little extensions to help them absorb more nutrients and water. They use active transport to pump minerals in. And so by increasing the solute concentration in here, that actually decreases the amount of free water in here, right? So there's less free water, which means there's a lower solute potential value. It becomes more negative. So we have a more negative solute potential inside of the cell, than we have out here. So it might still be negative out here, but it's higher than it is inside. Sorry, it takes a long time to write words like this. So out here, we have a higher solute potential. And remember, water is always going to move from where there's a higher potential to where there's lower. So the water is going to move in, as these red arrows are showing, through osmosis. So cells need to regulate the amount of water that they have inside of them. That's part of maintaining homeostasis. And there are a few different ways that they kind of balance the amount of solutes and the amount of water that they have in there, depending on the type of cell it is. Some animals live in seawater that's actually isotonic to the inside of their cells. And so they don't need to worry about maintaining that balance. Life actually evolved in the oceans, evolved in saltwater. And so a lot of living things just have that natural balance. That's why your tears are saltwater. A lot of the fluids within your body are actually salty, because that's where our cells are isotonic and can help kind of maintain that balance of water. Other organisms, like these little single-celled paramecium that you see here that live in freshwater, have evolved special features. You can see in this picture there are two things called contractile vacuoles. I'm actually going to show you how this works. There's no sound on here, but I just want you to take a look, and I'm going to skip ahead to one where you can see it a little better. So this paramecium, there are two contractile vacuoles right here and one over here. And you can see just pumped all the water out, then it slowly fills back up. Just osmosis naturally makes water move in. And then they contract that vacuole and actually pump that water back out. So these vacuoles just naturally fill up with water because of osmosis. And then after a while, they pump them back out to help maintain that balance. Plant cells usually exist in a slightly hypertonic environment, which causes the water, slightly hypotonic environment, which causes the water to move into their cells and keep them filled with water. That helps to hold plants up. That's actually part of their structure. Those cells need to be turgid. They need to be full of water. So that there's pressure on those cell walls, which helps keep the leaves standing up straight and the stems of plants standing up straight. If they're in an isotonic or hypertonic environment, those cells lose some of that water and become limp. That's part of why leaves look wilty if plants don't get enough water. And the cell membrane can actually pull away from the cell wall a little bit. You can see that in the picture over here on the right. That's called plasmolysis, and plants don't survive very well if that lasts for too long. So that's all on osmosis and diffusion and transport for now. Like I said, we'll come back to active transport in the next unit in trimester 2. A few reminders to kind of wrap up today. First of all, the Unit 4 resources are posted in the class materials now. This is a really important thing for you to be aware of and look at. I'm not sure how much people use these when you were in class, but especially now that you're at home all the time, it's important to realize these resources that are here for you. In those Unit 4 resources, you will find a copy of these slides. I know you have them printed out, but this is a good place to check if I add things to the slides. You can always go back and look at those slides that I added. Or if there are videos or animations or things that are linked to the slides that I show you during the lecture and you want to go back and look at them later, those links are in the note slides that are posted in the class materials. There are also 2 documents in there. One of them is called Prior Knowledge for the unit. Those are things that you probably learned in 10th grade biology, but you might need to review. So you can take a look at that and see if there are things that you need to go back and study that I expect you to know already. And there's the new content, which includes the learning targets and the vocabulary. So as you're studying and reviewing, that's a good place to go and just kind of use that as a checklist and see what things you understand and what things you might need to study more. Then a little bit about upcoming assignments and due dates. There's kind of a lot towards the end of this trimester because we added a couple extra labs in there. And I've kind of tried to space it out. So basically you have 1 assignment due every day for the rest of the trimester. They're not all super huge, and you can work ahead. So pace yourself, and I'm going to kind of tell you everything right now so that you know what's coming. Tonight, you have your last Mastering Biology assignment for the trimester due. There will not be a new one posted this week. This will be the last one. And tomorrow you have your ELISA post-lab questions also due. So both of those are things that you already had and we're already working on. So hopefully they're pretty close to done. You have a couple more things that I assigned today, and the order of the due dates for these has to do with when we're going to come back to these and talk about them in class. So by Sunday night, I would like you to read through the graphic novel and answer. There's about 6 questions about that I'd like you to have answered by Sunday night, because we will come back to some of those ideas next Monday. The part 2 of the osmosis and potato lab, you're going to go back to that now and look a little more closely now that we've talked a little more about osmosis and water potential. You're going to use some of those concepts to dig a little deeper into the data from that lab. So that assignment has been posted and that one will be due by Monday night. And by next Tuesday night, you need to finish up the cell size and shape lab that you did yesterday. Again, these are all topics that we're going to continue talking about into next week, but that's the order that they'll be due. Just for your planning ahead, there will be one more activity that's part of Monday's lesson. You should be able to get it done on Monday, but it will officially be due on Wednesday. And there will be one final College Board assessment that will be due on Thursday. I'll post it Wednesday morning, so you'll have 2 days to do it. But that will be kind of the last assessment of this trimester. And that will be all of the things that are assigned. What you see on the screen right now with those little notes on the bottom, that's everything that we have left for this trimester. So plan accordingly. Send me an email if you have any questions, or stop into a Google Meet. I'm having Google Meets from 9 to 10 in the morning and 1:30 to 2:30 in the afternoon every day. So drop in whenever you have questions, and I will see you tomorrow. Have a great afternoon.